Good morning. For me today is a great pleasure to be here and to be able to share my work and with the people in my lab. My name is Alejandro Reyes and my talk is titled Uncovering the Microbial Diversity through Metagenomics. As we all know, Colombia is a megadiverse country, and as you can see in the images, it has been widely recognized by its diversity in reptiles, mammals, plants, birds. But what do we know about the microbial diversity in Colombia? There's very little that we know about it. And how do we study that microbial diversity? Given that we have so many different environments, the best idea is to study that diversity is through metagenomics. Through metagenomics, we can analyze that microbial components of the microbial communities that are present in the different environments that we're looking for. But what does this have to do with bioinformatics? That is the topics that we are here, computational biology. So as new sequencing technologies have been developed, we have been seeing a huge increase in the scale of the throughput of the different sequencing machines with their corresponding decrease in costs. So this allows for getting a lot of data in a very cheap way. And then we get into the problem on how to do the data analysis. This slide, I brought it here just to show how it was before expected that a large percentage of the cost of sequencing was done into the sequencing itself. But nowadays, with the reduce in sequencing cost, now most of the cost goes into the experimental design and the data analysis. And that's where the bioinformatics goes in. It does require quite a bit of computing power and analysis to be able to perform these different metagenomic analysis. So in our group, we are the research group in microbial ecology and computational biology. And what we do is particular four main aims that we have. The first is to characterize microbial communities through metagenomics. That is a little bit of what I'm going to be showing today. But we also use computational methods for the assembly and annotation of different genomes. We develop methods for the study and characterization of viral communities, which is a very important work that we have been doing for a while. And it's also very interesting in characterizing that viral component of the microbial communities, but we're not going to be talking about that today. And also development of computational methods for the analysis and interpretation of different omic data. So today we're going to focus in this first point, characterizing the microbial communities through metagenomics. And in particular, we're going to focus on four main projects. The first project I wanted to bring here was developed by Andrea Borbon in our lab, and it focused on the cat microbiota of Andean bear. So we use metagenomics to do a surveillance of wild and captive populations. And why the Andean bear, or why the Andean bear is important. So the Andean bear is the only bear in a species of bear in South America. And it has a very wide altitudinal distribution range. It has been assigned as a vulnerable species according to the different conservation strategies. And this category of vulnerable has been done because mainly of habitat fragmentation and degradation and several negative interaction with humans. So as we moved forward and farming moves forward into their native environment, then the possibility for the bears and the environment of the bears seem to be fragmented, like by roads or by farming, and then there are more chances of interaction, of undesirable interactions with the humans. Importantly for this bear, even though like the other bears is mostly, has a physiology mostly of a carnivorous animal, their diet is mostly a plant-based diet. So most of their diet depends on different plant species that are found in the paramo and However, there are no suggestions of any adaptations to herbivory in their physiology. So that's where the gut microbiota becomes so important, because most of that plant um, nutrients that they acquire has to be degraded by the microbes. So that is crucial for the health and the immunity and the metabolism of the bear. So in general, a healthy gut microbiota will contain a balance between pathogens and symbionts, but if, with different environmental perturbations and reduced habitat quality, this can bring a gut dysbiosis where there will be an imbalance between those different microbial species, also leading to diseases like obesity, inflammatory disease, autoimmune disease, and susceptibility to infections. Given the lack of specialized mechanisms and structures for herbivory in the Andean bears, there is a high dependence on the gut microbiome for their health. And this means that this gut microbiome is also very sensitive to dietary changes and stressors. And here is the problem when we have captivity, because the nutritional management of the bears in captivity has a higher calorie intake that they usually take, and additional, they're supplemented primarily with domesticated fruits and cereals. That means that there is a change in their typical diet between the wild diet and now the diet in captivity. Additional, the habitat fragmentation and degradation change the environment, making it change the type of food that will be available for them to identify. That necessarily means higher diversity, higher dietary stressors, which could also lead to reduce microbial diversity. And that was our main question. We wanted to see what was happening both in captivity and animals in the wild. So we went in different parts of the Andes, in particular in close to Bogotá in Cundinamarca, and we sampled different animals in the wild and 2 different sites of conservation. And we characterized their microbiome, and the first thing we were able to see is that in terms of diversity, the wild animals definitely had more diversity than the ones that were in permanent captivity. Also, I want to point out here that we also had a set of animals, a pair of animals that were sampled over time, that they were captured as cubs in the wild and then brought to captivity. So they were raised in captivity. So we have that transition samples from the wild animals, those cubs when they were young, to their more adult-like in captivity. So when we look at both clustering and the distances in between the samples based on their microbiome, we can see that the wild clearly clusters separately from those that are permanent captivity. But those that were caught, we have some samples that cluster with the wild that happen to be the very early samples. And we can see at least 2 samples clustering in the wild, but then they very fast move to look more in their composition to their permanent captivity animals. So we look at the compositions, we can see a very drastic difference between, in particular, the composition here of different protobacteria and the Bacteroidetes that we have here in the wild compared to those in permanent captivity, which has a very large increase in Firmicutes. Also, the wild-caught had a very intermediate phenotype in which they have a little bit of the composition of the ones in the wild, but they are decreasing their Bacteroidetes, also and increasing the Firmicutes. So we have in general a dominance of Proteobacteria and Firmicutes, in particular Clostridiaceae in captivity animals. When we look at the shared microbiome, it's interesting to see that most of the bacteria are shared between all animals. There is really no species that is unique to either the wild-born and later captured cubs or the ones in permanent captivity. And the highest shared number is precisely between the wild and the wild-born, suggesting that these wild-born animals still maintain some of that diversity that the wild animals had. There's some unique OTUs for the wild, suggesting that the wild still have more diversity than the captivity ones. And those are the key microorganisms that are being lost as the diversity decreases in the captured individuals. So when we look specifically at the cups, we can see in terms of the distance to the wild animals, to the captive animals, that distance is decreasing over time, suggesting that they're converging more towards a captive-like community. And the number of OTUs shared with wild individuals is also decreasing, suggesting that they're diverging more from the wild individuals. So this is very important for the bird conservation, and it should lead us to think how we're giving and how we're treating animals in captivity, in particular if we want to return them into the wild. The next study that I want to show you was performed by Elena Quintanilla, and it was precisely looking at the corals. So corals is a very different environment and a very important environment in the Caribbean. And we know that climate change have brought increases in temperature, and we have all seen the decline on the coral ecosystems. So there was this disease that was named as necrotic patch disease, where the corals start dying in patches. So we wanted to see if this was associated with some microbial change. So we wanted to characterize the microbiome of healthy individuals in two different sections, and in diseased individuals in a healthy patch and in a patch very close to the necrotic patch. So what we can see just in terms of the shared diversity is that when we look at all the healthy individuals, the diversity is conserved. But when we add the diseased individuals, shown here in orange and yellow, the yellow one being the ones that are very close to the necrotic patch, and the orange one, distant one, we can see that the distant is healthy and clustered with the healthy, but the yellow that were close to the necrotic patch is really changing their composition. So when we look at the composition, we can see that there seems to be a very stable microbial diversity that doesn't have a very high diversity in terms of microbial species, some with low abundance, some with higher abundance, and is particularly dominated by Endozoicomonas and bacteria from the genus Mycoplasma. And there was an interesting inverse relationship where the more abundant Endozoicomonas, the less abundance the Mycoplasma, and the opposite was also seen. The ones that had higher abundance of Mycoplasma had less abundance of Endozoicomonas. But this is the characterization of all the healthy ones. When we added the diseased ones, you can see that there's a whole lot of other OTUs that now begin to appear that are not causative of the disease, because they're also present in the other region, but they're enriched. Definitely potential opportunistic pathogens that are being enriched by the modification of the environment. And we can clearly see a decrease in particular of Endozoicomonas and in some of the Mycoplasmas. So we can definitely see that even though it doesn't seem to be a microbial caused disease, there's microbial changes, and these microorganisms could work as markers to be able to follow the disease and identify different corals that could be at risk or under stress because of changes in their environment. A different study, switching topics quite a bit, is this one here about the microbial diversity during the fermentation of cacao. This was performed by Mauricio Pacheco in the lab, and this is also very important for country, because in Colombia, we're beginning to grow, as we grow coffee, we're beginning to grow cacao to make high quality cacao. So we went to different regions, in particular here Santander and Antioquia, to sample coffee, cacao beans during the fermentation process. So in these boxes, you can see the mixing of the box during the cacao fermentation. So this was followed over time over the fermentation process in hours in two different regions, in Santander and in Antioquia, and in two different times of the year, during the dry season and the wet season. And what we can see here is really a variation in the microbial community, as we will expect. So here we have the yeast, here we have the bacteria. This is the relative abundance over time for each of the two regions. each of the two time zones. We can see that the process is really consistent and is very reproducible in the different regions. And what we see is a very high domination of Hansenísporia as the yeast. And if we were able to compare the yeast and the bacteria, we will see a high abundance of yeast early on in the process, along with the Enterobacteriaceae. That this will generate the right substrate so that then acid lactic bacteria like lactobacilli and fructobacilli will begin dominating and will begin changing the environment, generating the byproducts that then the acetic acid bacteria like Acetobacter and some bacteria from Acetobacteriaceae will take and will dominate to finish the process. We were able to see some characteristic, for example, in this process, we see that there was like a delay or some changes in the process. And this was due to mixing of the cacao beans during the fermentation in the box at not the right time. So this caused a delay in the fermentation process. So you can see by monitoring the microbial diversity, how specific and how well that fermentation process is going. And then we can see at more detail. So we did oligotyping on these samples, and each one of these rows correspond to different oligotypes for each of the Enterobacteriaceae, the Lactobacilli, the Acetobacteriaceae, and so on. And then we can see that in many moments, we have a high diversity of different oligotypes that are quickly reduced to one dominating oligotype. And this particularly is the case, for example, for Acetobacteriaceae, that we know that at the beginning of the process where Acetobacter appears, it seems that there's a lot of different oligotypes. And as the process continues, then only one of those oligotypes is selected. So it's interesting that it's exactly the same oligotype in the different regions in the different times of the year. So it seems that there's an innate diversity of different oligotypes that are present in the boxes, in the tools, in the samples, but then the environment and the conditions quickly select for a given oligotype. And this could be very important because based on this, we can take the most proficient oligotypes, culture them, and use them as a potential starter culture in order to better standardize and better perform the cacao fermentation. And lastly, the last project I wanted to show was this performed by Carlos Posada. Now this is in water. So we have shown a little bit of animals, conservation, corals, environment, fermentation, food security, and now waters. So this was analyzing the microbiome and the resistome, in particular of residual waters and the Bogotá River. So the Bogotá River is the main river crossing at the side of the city. Bogotá is a very large city in Colombia, the capital. And we decided to sample at the birth of the river, right before getting into the city and at the end of the city, together with 3 hospitals that discharge their waters into the sewage system that eventually leads into after passing through the treatment plants into the river. So when we look at the diversity of these communities, we can clearly see that the river at its birth has the highest diversity. And then we have some of the samples from the hospitals, some of the samples from other hospitals here. So in general, what we see is that there's so this is showing one sampling that we performed in 2015. This is a sampling that we performed in 2016. And what we see is that rivers in general, even especially the early sites of the river, the birth of the river and before the city, has higher diversity than what we can see in the residual waters. And then when we look at the compositions, you can see how different is the low river that is at the birth of the river, that is very, very different. Sorry, this here, the medium and this low. This river is very different to the composition of the hospitals that you can see here. So the hospitals in general then have to have a very high increase of Bacteroidetes and Firmicutes, which, if you may know from other studies, these are the 2 main components of the human gut microbiota. So this means that those residual waters are very loaded with probably fecal contamination. But importantly, what we see is that the river, after passing through the city, begin looking with that increase in Firmicutes and Bacteroidetes similar to those residual waters. When we do the clustering, this is based on the 16S, and this is based on the Resistome, we can see that some of the samples from the river, especially the low river that is after passing through the city, begin to cluster with the hospitals more than the waters. And interestingly, in terms of the Resistome, more of the river begin clustering with the samples. So in the Resistome, we were able to assemble different resistance. We can measure the abundance of those resistance. So we noticed that most of the hospitals have a lot of resistance genes in those samples, but also the low river begin having a lot of resistance determinants as well. And we can see there's a whole wide diversity of resistant genes that we can find in all the samples. And it is known that we can find resistance upstream in pristine waters, and this is just based on the natural resistance that are present on the different microorganisms. But the resistance that are found downstream, those is mainly based on resistant genes that can be easily transferred between the different organisms. So this is very important for further studies. So this was a very brief presentation showing just for projects that I wanted to bring today. So in summary, we can say that the microbiota is selected by the organism and the project process, but it can change in abundance rapidly by the changes in the environment and diet. That bacteria has co-evolved with the host, in particular in the cases of the berries and the corals. And similar strain and species can be found associated to the same animal and process throughout the country, as we saw both with the berries and with the cacao fermentation. Changes in the microbial community are also strong biomarkers of disease, like we saw with the corals. And sometimes we need to look deeper, probably at strain level, to uncover some of the potential of the microbiota, like we saw with the cacao fermentation. Also, changes in the microbiota, even in a single member, can have a cascade effect that could impact positively or negatively a community and its environments. Finally, it's important that we think that we as humans, we have a significant impact on the environment that surrounds us, and in particular, the microbiomes. So as we saw with the use with antibiotics. So this is a call for very responsible use of those antibiotics and to have in mind the potential impact that we're having. Finally, I want to thank you for your attention, and I want to thank everybody in the lab. So this is our group, and I just show you the work that was done by 4 of them, but there are many other works that due to time and I wasn't able to show here, and thank you very much.